Acute-phase reactants are soluble proteins that belong to the innate immune system, quickly increasing in response to inflammation or infection to help protect the body. They rise rapidly in the body during infection, inflammation, injury, or tissue trauma, acting as early responders to help the immune system manage damage and fight threats. Once inflammation begins, their levels start to rise within 12 to 24 hours, signaling an early immune response. These proteins are produced mainly by hepatocytes in the liver. Their production is driven by inflammatory cytokines like interleukin-1, interleukin-6, and tumor necrosis factor alpha, which signal the liver to begin acute-phase protein synthesis. These cytokines are released by monocytes and macrophages right at the sites of inflammation, sending signals to the liver to produce acute-phase proteins. Many acute-phase reactants work as opsonins. They attach to microorganisms and make them easier for phagocytes, like neutrophils and macrophages, to engulf and destroy. CRP, or C-reactive protein, was first discovered in 1930 in patients infected with Streptococcus pneumonia. It got its name because it reacts with the C polysaccharide found on the pneumococcal cell wall. CRP works a lot like an antibody, but it's actually part of the innate, non-specific immune system. CRP needs calcium ions to bind antigens like phosphocoline on microbes. Without calcium, it can't attach properly and carry out its immune functions. CRP can bind to several targets: to phosphocoline on bacterial and fungal membranes, damaged host cells, and even some parasites. By attaching to these targets, CRP acts as an opsonin, marking them so neutrophils and macrophages can easily engulf and destroy them. CRP can also activate the classical complement pathway by binding to C1q, working in a way similar to antibodies to help clear pathogens. CRP responds quickly. Its levels rise within 4 to 6 hours after infection, inflammation, or tissue injury. During acute inflammation, it can increase 100 to 1,000 times above baseline, usually peaking within 48 hours. CRP is the most widely used lab marker for acute inflammation. Because it rises so quickly, it's a very sensitive indicator, showing changes in disease activity almost in real time. Serum amyloid A, or SAA, is another major acute-phase reactant, ranking second only to C-reactive protein in its response to inflammation. SAA levels can increase up to 1,000-fold during infection or tissue injury. In the blood, SAA binds strongly to HDL cholesterol. HDL then transports SAA to sites of infection or inflammation. SAA doesn't just act as an acute-phase reactant, it also behaves like a cytokine. It helps modulate the immune response by recruiting immune cells to the site of infection or inflammation. Serum amyloid A can activate monocytes and macrophages. Once activated, these cells release pro-inflammatory cytokines like interleukin-1, interleukin-6, and tumor necrosis factor alpha, which amplify inflammation, recruit more immune cells, and boost phagocytosis to help clear pathogens. SAA levels rise higher during bacterial infections compared to viral infections, making it a useful marker for distinguishing the type of infection. Serum amyloid A can accumulate in atherosclerotic plaques, connecting chronic inflammation to cardiovascular disease. High levels of SAA in the blood often signal ongoing vascular inflammation. Alpha-1 antitrypsin is the main protein found in the alpha band during serum protein electrophoresis. Alpha-1 antitrypsin AAT acts as a general plasma inhibitor of proteases, protecting tissues by neutralizing enzymes released from leukocytes. During inflammation, neutrophils release elastase, an enzyme that breaks down elastin and collagen. If this process isn't properly controlled, it can damage surrounding tissues. Alpha-1 antitrypsin counteracts neutrophil proteases, helping protect lung tissue from damage during inflammation. AAT also helps regulate pro-inflammatory cytokines like TNF-alpha, IL-1 beta, and IL-6. By keeping these signals in check, it limits harmful inflammation and protects tissues from damage. AAT deficiency can lead to premature emphysema, especially in smokers. Without AAT to control them, proteases run unchecked, destroying lung tissue and potentially causing emphysema or pulmonary fibrosis. Haptoglobin, HP, is an alpha-2 globulin seen on serum protein electrophoresis. As an acute-phase protein, its levels rise during inflammation to help the body respond to stress or tissue damage. Its main job is to bind free hemoglobin released during hemolysis, protecting the kidneys and preventing the loss of iron from the body. Free hemoglobin released during hemolysis can catalyze reactive oxygen species, ROS, formation, causing oxidative damage. Haptoglobin binds free hemoglobin, forming a complex that is cleared by the liver. This prevents oxidative stress and protects tissues, especially the kidneys, from damage. Fibrinogen is a soluble plasma glycoprotein and an acute-phase reactant. Its levels rise during inflammation. In coagulation, thrombin cleaves fibrinogen, producing fibrin monomers. These monomers polymerize into fibrils, forming the structural framework of a fibrin clot, essential for hemostasis. Fibrin clots not only strengthen a wound, but they also encourage endothelial cells to stick and grow, helping repair and restore the blood vessel lining. Ceruloplasmin is the main protein in plasma responsible for transporting copper throughout the body. Ceruloplasmin also acts as an enzyme, turning toxic ferrous ions, Fe2+, into the safer ferric form, Fe3+, helping prevent oxidative damage in the body. A depletion of ceruloplasmin is observed in Wilson's disease, a genetic disorder. In Wilson's disease, copper accumulates in the liver and other tissues like the brain, corneas, kidneys, and bones.